Good afternoon, everyone. Welcome and thank you for joining us for the second Infectious Diseases Across Scale seminar for the semester. It is with great pleasure that we welcome Dr. Charles Nunn. Dr. Nunn is a renowned evolutionary biologist whose interdisciplinary research transcends traditional boundaries, merging evolutionary medicine, disease ecology, and global health to shed light on infectious disease dynamics and emergence. Dr. Nunn earned his bachelor's degree from Dartmouth and went on to obtain his PhD in biological anthropology and anatomy from Duke University, global level. Afterwards, he carried out postdoctoral work in biology at the University of Virginia and in evolution and ecology at the University of California, Davis. From there, Dr. Nunn went on to the University of California, Berkeley as an assistant adjunct professor in the Department of Integrative Biology, and before joining Harvard's Department of Human Evolutionary Biology as an associate professor. After his time at Harvard, Dr. Nunn returned to Duke, global levels, in 2013, when he is full professor in the Department of Evolutionary Anthropology and the Duke Global Health Institute, during which he taught me Bayesian statistics. Dr. Nunn employs an interdisciplinary approach to advance evolutionary perspectives on societally important questions, including One Health and pandemic prevention. His work in Madagascar is investigating how land use change and market integration influence infectious disease dynamics in humans and other animals. Moreover, Dr. Nunn's group is also investigating zoonotic disease emergence and the predictors of cross-species transmission in primates and other animals. Dr. Nunn's dedication to scientific inquiry and his commitment to addressing real-world health issues make him a distinguished leader in infectious disease dynamics. We eagerly anticipate his enlightening presentation and the valuable insights he will share with us today. And without further ado, please join me in extending a warm welcome to Dr. Charles.
Okay, thank you very much. That was a nice introduction, and it's great to be here. Great to meet a lot of new people and to reconnect with some others that I've known for many years. And so thank you very much for this invitation. If we think about the zoonotic disease pandemic life cycle, let me see if I can get rid of that. Well, maybe I can't. We'll just have to live with it, I guess. If we think about the zoonotic disease pandemic life cycle, we can think about spillover of a pathogen from some animal into human populations, the transmission of that pathogen locally within a community, right? Possibly inter-community transmission. That's a requirement for this to go global, when it might finally land in a larger city with good transportation, airplane flights, things like that, where it can eventually go global. And today I want to talk about the work that we're doing in Madagascar, focusing on the early stages of this cycle, this pandemic life cycle. And I want to tell you a few things. I want to show you how network approaches can be useful for thinking about these questions at the human-animal interface within villages, among people within a village, and then also between communities, so we can look across scales with these network approaches. I also want to really emphasize that this part of the story is really essential. We have to have that connectivity among different communities, usually for a pathogen to land in the places where it can really disseminate more widely, globally, potentially. And then finally, I want to convince you that we really need to understand not just phenomenologically what these networks are, but why they're created. You know, why are people connected? Why are certain people connected to animals? Why are certain people more connected to other people? You know, to really understand these networks and to understand what generates that connectivity. So I work in an amazing place. I feel very fortunate to work in northern Madagascar around a major national park, Marijeji National Park, where there's incredible biodiversity. It has a big altitudinal gradient. I said a major national park, but it actually only gets about 1,500 people per year. It's really pretty remote, a beautiful location. But most of the time, and of course, there's all kinds of incredible biodiversity in Marijeji, like the helmet vongas, these silky shafaks. And I actually took this picture of, I don't know if you can see it, but this is a leaf-tailed gecko. That's one of my favorite organisms in Madagascar, and especially Marijeji. And we, of course, spend most of our time outside this national park. I usually find an excuse to get in there, and it's certainly what drew me to this area. But we work in these villages around the national park. And first, I'm going to talk about these three villages where we started our work in Datsakala, Mandena, and in Sarahandranu. and just give you a little introduction to what life is like in these villages. This is one of those villages, very typical kind of scene. These are farmers, smallholder farmers, for the most part. You can see the agriculture goes right up to the edge of the national park, very typical kind of situation where there's a lot of land use change that's been happening and is still happening in these communities. So no running water, no electricity, very, kind of simple village life. They live in these villages and then go out into the fields during the day to work in the fields.
Hold on just a second.
Oh, sure.
They're not seeing the slides move.
Oh, no.
It says the screen sharing is paused.
Okay. Oh, let's get rid of this thing first of all. And maybe this. Also, okay, now I should do that. Let's hit, it looks like it's sharing.
Well, yeah, it changed. Now it says sharing. So maybe
We good?
Okay. we're
Okay.
good now. Yeah.
Okay. This is Madagascar, so it's rice farming. Okay, so these are mostly rice farmers. Almost everyone has a plot of land where they're growing rice. They're doing this by hand. For the most part, some of the milling of rice, some families have, pay for it to be milled. But mostly they're doing this by hand. They're carrying this back to the village on their backs. There aren't really beasts of burden. There's no tractors, things like that, at least in these 3 villages. They cook with wood, so very, kind of basic cooking, not charcoal, not gas, using wood, not electricity.
I don't think it's progressing.
No, it's not. Okay, you see there's something going on here. Okay. Let's see. It says it's sharing there. Should I just stop it and start it again?
Try to hear what we're gonna stop instead of just presentation.
Sorry about that for those online. Let's see, it looks like it's sharing, doesn't it? Oh, it's paused again. Oh. I wonder why that's doing that. It says it is there, but then is it something about, I could do it from a PDF instead of Keynote? I don't know if that would make a difference.
That might be the difference here.
Or maybe share the whole desktop.
Okay, that's an idea too. I like that. I like that idea. So share the whole desktop like this, right? And then go back to Keynote. And now it looks like that's staying green. Okay, well,
There we go.
here for those of you online, here they are harvesting the rice, cooking dinner. And then this is where I left off. And I hope everyone knows what this is, vanilla beans. And I brought some vanilla beans. I always try to bring vanilla beans for my coffees. And so please do, I don't know what to do with all these vanilla beans. So please take a vanilla bean if you would like a vanilla bean. So you guys on Zoom should have showed up today. I always end up with so many kilos of vanilla beans, and so I always bring them to my talk, as long as I have some. So please have a vanilla bean, if you would like a vanilla bean, or two. This is the big cash crop in the area. Any decent farmer is going to have some vanilla plants somewhere on their land, sometimes hundreds or even thousands of these plants. And it's really an important crop for the people there. And vanilla and other cash crops like coffee, cloves, other kinds of things are really fueling a lot of market activities and enabling people to buy things, like this satellite dish you see on that home. And so a lot of what we do in our work there is consider market integration. And that's this transition from a subsistence lifestyle to one that's more market-based, so that they're selling things onto the market and using that cash to buy things that they want, okay? And so I'll talk a little bit about that today. So I hope that gives you a sense of, the life in these villages and the populations that we're working with, and we do work closely with them to identify the questions that we pursue in a kind of participatory research fashion. And that includes asking them about the health problems they have. And so, the work I'm going to talk about today represents many years of building these relationships and talking to the people and, having that help guide the research that we do. Okay, so I want to talk about this work. Oh, there's my copy. I want to talk about this first major project that we landed, this first major grant that we landed to fund the research in Madagascar. It's an ecology and evolution of infectious disease project funded by the NIH on the impact of land use change on transmission, potential networks, and disease spread in Madagascar. It's a very interdisciplinary group of people. I don't think anyone in this room would know all of the people on this grant. We have a mathematician, a sociologist, our colleagues from Madagascar, our scientific partners, and economists. Hillary Young, some of you might know Hillary, a disease ecologist, Pablo Tortosa, Shai Philosoph, another disease ecologist, all working together on this project. And the project goals are to generate transmission potential networks that represent hypothesized pathways of infection between humans, other animals, and the environments we share. And I should say this grant is wrapping up. We're in kind of the final months of it at this point, and we're getting a lot, we have all the data and we're getting a lot of analyses done right now. So I'm going to share some of those results with you. So we generate these transmission potential networks. We can then assess whether they reflect actual patterns of infection with micro and macro parasites. Okay. And then very importantly, we also, with our colleagues from the social sciences and some of my background in evolutionary anthropology, human behavioral ecology, we also want to investigate the socio-demographic and behavioral predictors of contact patterns, really trying to understand what drives this, right? And I put that in as a different color because I think that's a really important, thing to achieve, and it's a part of our project because, of course, this offers us opportunities for some kind of intervention, if we wanted to change patterns in the event of an outbreak of some kind. And so I'll talk about the work from a bunch of trainees on this project. Georgia Titcomb was a postdoc on the project. Now she has an assistant professor job at Colorado State. Kayla Kaufman at UCSB, 4 people from my lab, Charles Chu from Duke NUS, and our project manager, Michelle, who's involved in all of this. So these are projects that most of the projects I'll talk about were led by one or more of these folks. Let me tell you a little bit about the data we collect. First of all, we have human survey instruments, working with our colleagues, the social scientists to develop these with focus groups and all kinds of other things to really fine tune the questions. We collect data on social relationships, socioeconomic and demographic data. We ask questions about animal interactions, health and symptoms, and then various measures of market integration, like, how many items they own that could only be purchased from the market, like cell phones and radios and, things like that. We also collect GPS data on people. We give them these data loggers. They record their location every 3 minutes. And so we get really rich data on where people are going and also how often they're in proximity to other people, right, who are wearing these devices. And of course, we also give them to animals. So we give them to the cats, the dogs, the pigs, and the cattle in these systems. Okay. I should say that people wear those devices for about a week. They can wear them up to 6 weeks, though. They wear them at least 1 week. We also get fecal blood and urine samples, and you can see our sample sizes here as well. I should also say we're only looking at adults over 18 in the studies I'm going to talk about today, but we're going to be expanding to look at children as well. Then we take these samples, we identify parasites, usually using metabarcoding approaches, various kinds, but other approaches as well to identify infectious disease in these samples. We also do some serological work looking for antibodies. We do a lot of small mammal trapping, thousands of animals trapped under this first grant. We collect a lot of invasive species. There seem to be an endless number of black rats in the system that we can't deplete no matter how hard we trap, it seems. We set up 11 by 11 grids and trap over 7 nights. We also get a number of the endemic species, and we trap it across different land use types, including in the villages, but various types of crops, brushy regrowth areas where land is fallow and plants are growing up again, secondary forest, and into the national park itself, where we actually also do transects up to the summit of the mountain, the main mountain in the national park. We also trap bats. We've got about 500 bats that we trap in these villages as well. So a lot of data on the small mammals. We use satellite imagery, satellite data We classify it into different land use types. As you can see here, where it's classified, these 30 by 30 grid cells are classified into village, rice, forest. You see kind of some of the ways we deal with the arbitrariness of where the grids are placed is where it looks a little fuzzy around there. That's a technique we're using just to deal with the uncertainty in these associations or in these classifications. And then, of course, we can then take the GPS data that we get, whether it's from an animal or a person, and look at where that person is going. This is a utilization distribution based on their GPS data. And we can say, okay, they're spending, a lot of time in this rice field and a lot of time in the village. And this just shows, kind of a video of 3 different individuals, a dog and 2 people, and how they're moving over a very short period of time compared to what we have. And you can see, we can notice times when they're in proximity to one another, right? Those 2 people, and when they're in proximity to that dog out in the rice field. So one of the things that's been great about working with, Shai Philosoph or Peter Muka, the mathematician, or the sociologist as well, are all the different ways we can bring network perspectives into this work. One of the things we do a lot of is build these kind of environmental networks. And so we have a bipartite network where we have people in the different grid cells that they go to, okay? This is one person, I showed this before, and the different grid cells that he or she went to. And we can, of course, expand this out to all of the people in a village, right? And all of the grid cells that they go to in this bipartite network. And then we can get a projection of that shows how people are just connected overall, right? How much do they overlap in their ranges just in general? Or we can say, how much do they overlap in rice fields? Or how much do they overlap in secondary forest? Right? And so if we have hypotheses for certain organisms transmitting in certain environments, we can ask, do these people seem to have sharing of those environments? If they do, then we should expect to see that if one's infected, the others will be infected as well, right? So this is a very flexible approach that we can use in all sorts of different contexts. So we have the proximity data, but we also have this kind of environmental overlap data that can be looked at in many different ways. And then we also have social network data, because we ask them questions about who do you hang out with, who do you go to for help when you're forced, who do you borrow money from? And these are really intended to capture deeper relationships, and kind of time that might be spent in closer proximity that we might not catch, with the snapshots of the GPS data and things like that. And this was actually some earlier work we did done by Ajale Owens, who was here at Emory after she graduated from Duke, and has since graduated. She got her MPH here. So just in case anybody knows Ajale. Now we have, we find all kinds of viruses, bacteria, protozoa, all kinds of things in these populations. I'm just going to give you a snapshot of some of them. We just have a paper coming out right now on hantaviruses found entirely in the rat population, especially in rice fields, paramyxoviruses, astroviruses. Another paper we published a lot of leptospira. And we find this in the small mammals as well as in the bats and in the domesticated animals and in the people. In terms of kind of worms and protozoa, pretty high prevalence of hookworm and dog hookworm, canine hookworm, various other organisms, blastocystis, wide range of eukaryotic parasites in domestic species and the small mammals. And then we have a lot of data on ectoparasites, and this is where we've done the least work so far. We have a student actually from Sherry Briggs' lab who's doing a lot of this analysis, and it's leading to some interesting initial results, but I'm not going to talk about that today. So obviously this is too much to talk about. I can't look at all of these with networks today. I'm just going to focus in on 3 of them where we have kind of good prevalence data to work with these network approaches, the 2 hookworms and blastocystis. Okay, so a lot of you, probably know about hookworm, human hookworm, most common hookworm species globally. It's got fecal transmission. The larvae penetrate the skin, right, and have this, they get up to the lungs. You guys probably know this, and cough them up and then swallow them. That's how they get into the gut, and then the eggs get shed out. So it's kind of an interesting organism, but you get it from walking in the soil where these worms are, or the larvae are. It has sensitive environmental stages. It requires a shady, or it benefits from a shadier, kind of moisture environment for hatching. And then we have this dog hookworm, Ankylostoma silenicum, pretty high prevalence in people. So right off the bat, we think, here's good evidence for a zoonotic organism, affects dogs, cats, and people. Prevalence of dogs in the study region is about 73%. So these poor dogs have a very tough life, I have to say. And they get a lot of hookworm. And it's also soil transmitted. So first of all, one of the things that we've noticed with all of our GPS data is that use of forest fragments is a good predictor of hookworm prevalence. And it kind of makes sense because it's a place where people might go to defecate, right? And it's also got that moist, shady habitat. So we had kind of two dominant hypotheses. It's either transmitting in the villages around places where latrines might be, or it's out in other areas where people might be defecating, in the open essentially, but in a place where they have some privacy. And so this is a forest fragment use, and this is kind of the number of grid cells that a person uses. It's a measure of their intensity of that. I'm sorry, it's a measure of the intensity of the forest fragment use, hectares per week. And you can see that there's an association there. We can also develop build these models that look at how people overlap in the same forest fragments themselves. Okay, so this is a network now using the environmental data, showing how people overlap in in particular grids in our habitat. And we can test whether that predicts patterns of infection. And for this, we use this network K test. It's a permutation test that assesses whether the contact network that you have represents a transmission pathway. You calculate the mean number of cases, or K, within one step of an infected case, and then you randomly reallocate those cases throughout the network and compare the observed K to this distribution of random Ks. And we find that for two of these villages, the secondary forest network predicts infection, okay? So this shows the observed value and the kind of distribution of values used, generated through this permutation process. And we find that there are 23% more infections along secondary forest edges from infected nodes. So it's kind of a measure of that risk, right? If you're connected to someone on that network who's infected, right, you have a 23% higher chance of being infected. And it's even stronger in this other village A, 47% more infections along secondary forest network edges. Okay, we find that other variables, the nice thing is we have all of this other data that we collect through those survey instruments. And we find that education is also a predictor of infection, as you can see here. Lower education, right, Nunn, primary or secondary, has a higher risk of infection. And we see a sex difference. This sex difference is generated because men are more often out in these fields and presumably using these secondary forests, as I showed in that previous slide. So men are twice as likely to be infected as women, and men have larger home ranges and use these forest fragments twice as much as women. And then the other main result we find is that owning a dog is also a predictor of infection, okay, with the ankylostoma, with the dog hookworm. That's the main variable that predicts infection for that hookworm. Now, you might have noticed I talked about two villages, and you might remember that we have three. And village S, one of these villages, is different. And we puzzled over this. I mean, here's the results from these other two villages, showing that sex difference, showing, in this case, home range, focused on those secondary forest plots. Village S, we don't find that. This puzzled us for a while, but looking at the situation in Village S is, I think, informative. Here are the other two villages. You can see, the village here in red. You can see the kind of rice fields. There's a lot of forest around these rice fields, right? As you can see, Village S is a different situation. It's just huge rice paddies with no kind of forest edges around them. And so, the interpretation we have is they're not going in, they don't have forest edges to go into if they have to use the bathroom, right? If they have to use the toilet. And instead, the risk we expected would be concentrated in the village. And in fact, we have, the kind of data, we have other studies we're doing. This is by Kayla Kaufman, showing how people are using the different habitats in these different villages. And so for Village S, there's much greater use of the village, as you can see here. About the same for the rice fields, maybe even a little bit more for some people, but the secondary forest that exists is not used as heavily in this population, this other village. And instead, if we build a network now from the houses and how far apart the houses are, so we have an edge here, if a house is within a certain distance of another house, okay, it might be 10 meters, it might be 50 meters, we do it with different values here. But regardless of what those values are, the house network itself is a predictor of hookworm infection. So it's suggesting there's something that's happening kind of more locally in the village that's influencing the exposure to hookworm in this other population. Okay, so in this case, secondary forest is not predictive, but the household network is. So it's just kind of an example of how we can use, the different network data we have, including where their houses are, right, to say something about or to infer something about the transmission of this organism in different environments. Okay, so for the overall, an overall conclusion here for the hookworm story is we have two villages. In two of the villages, the primary route of transmission appears to be via open defecation away from the home in these agricultural fields with increased parasite survival in these shady areas. You can maybe see this, rice field and the kind of forest that surrounds it. Networks provide new insights to the landscape of infectious disease, right? We can predict the patterns of infection and even get some new insights to how that might vary in different villages, depending on the environment and the habitats in which people are living. And we don't actually have to sample the environment to do this, right? We're just doing this from the network data itself. And zoonotic infections are also occurring with variation across villages. Now, we also have looked at blastocystis, which is kind of an interesting organism in a lot of ways. It's a protozoan. We have pretty high prevalence in this population. It's one of the most common parasites globally. Some would say, at least some of these kind of strains, variants of Blasto are probably not even parasitic. They might be commensal, okay? There's a high diversity, a wide host range, and the strains vary in pathogenicity. It's transmitted via fecal oral route, and multiple transmission pathways are possible. And we had hypothesized that it could be socially, through kind of, rich interactions, time you really spend in real relationships where you might be eating and drinking with people, right? Those social relationships. It could be through shared food and water within the villages, again, kind of based on the spatial proximity of houses, or it could be zoonotic. And so we can use network approaches to test predictions for these different transmission potential pathways. And the first result to say is the friendship network is not predictive at all. In fact, we haven't found that this is predictive of much. Kind of sadly, you would think it would have been, but we are not finding that the friendship network is predictive. We use that K test, but we're also using some other tests, in this case, kind of network autocorrelation approaches, where you can look at the proportion of infected alters, the proportion of others you're connected to that are infected, okay, as a variable in the models. And that turns out to be, oh, sorry, I jumped ahead of myself. In contrast, we find that the village itself and that village proximity really explains a lot of the infection patterns that we see. So if you're living within proximity to another household where someone's infected, your probability of being infected is higher. Okay, so that's true with the K test. And if we look at this proportion of infected alters based on that household network. This is actually kind of an interesting village if you're interested in what's going on with this network. It's actually two villages that are a little bit distant from each other. Actually, I'm not sure why it came out that way. I think, I'm not sure what the orientation of it is. Now that I say that. But anyhow, the household network itself is what's predictive. And when we look at both of those variables in the same model, what we find, we find that proportion of friends infected is not informative, proportion of close neighbors infected is, and none of the other variables are significant in these models that Tyler did in this paper we published recently. And some of the other work that Alma's done with a larger sample size, she does find that hand washing is a predictor of infection. And that's washing only with water gives you a higher risk of infection as compared to using soap. Now, in terms of the animal contact, that third possible pathway, we don't find any evidence that animal contact was significantly related to infection. This is self-reported contact with different animal species. In addition, of the 133 domesticated animals that we sampled, 20% were infected. However, the owners and their animals always had different subtypes, and mostly these animals had different subtypes from all that we found in the humans. And then of 856 of the rats that were sampled, 5% were infected, but for a subtype not found in people. So it really looks like it's something about the local, again, like that hookworm, like the hookworm example I gave you in that one village, it's something about that local environment where people are living that's really predictive of infection for blastocystis. Okay, so across the 3 villages, blastocystis spread is most likely spread through environmental pathways within villages rather than through social or animal contact. These pathways probably involved shared food and water sources, near people's homes or fecal contamination in the environment by infected individuals. And we find no evidence of sharing subtypes between humans and other animals, suggesting that this zoonotic transmission, if it's occurring, is rare. At least we didn't detect it. Now, one of the exciting things here is we have all this data on the animals as well. And so this is some new work that Lev Kolinsky and Camille Sisto in my lab have been working on. They've been doing, trying to build in the animals into these networks, more formally. And they're doing this in 2 ways. One is to use the survey data, because we talk to these people, we ask them, do you have, interactions with, what animals do you interact with? What kinds of interactions do you have? Have you, cooked one of them? Have you been scratched or bitten by one of them? Have you raised one of them? Do you have them inside your home sometimes? So we can ask all of these questions. We identify some of these as higher risk. Those are the ones with the asterisks by them, higher risk for zoonotic disease transmission. That we can, use these data and build bipartite networks again. Looking at, this is self-reported, each individual, who are they, which animals are they interacting with, and what kind of interactions are they having? And so this is one of those kind of networks, showing, it's kind of, I know it's kind of a weird network because it's a bipartite network, but, represented in a different way than the other ones I showed you, with poultry and cows and cats being some of the most, kind of common interactions, as well as rodents. And then these are the predictors of the edges on this network. What predicts the interaction, right, between people and these different animals, okay? And so we use what's called an ergom, or an exponential random graph model, to predict those edges based on characteristics of the people and also characteristics of the animals and various kinds of interaction terms that we might want to have in this model. So the main thing to share with you is that, these early results, it looks like land size is a predictor of interactions, probably represents something about agricultural wealth. So people have more agricultural wealth are reporting more animal interactions, and that might make sense. They probably own a lot more animals. As well, men are reporting more than women, and age has a negative effect. So mostly younger men. In addition, there's some interaction terms that are interesting. I didn't want to put everything up here on one slide, but those with more wealth are less likely to report contact with wild animals. So it's suggesting that maybe some of the hunting of wild animals that's going on is related to people needing that protein source and perhaps pursuing that if they don't have other access to food and protein. So that's the survey data. But we can also use the GPS data, of course. And here's an individual with his GPS device around his neck. In this case, we're only looking at the spatial contact, the environmental overlap. We're not looking at the close contact, the being in proximity at the same time. It's just shared habitat use. And you can see just the sample sizes we're dealing with here. This first field season was cut short because of COVID. And so these are the 2 full seasons we had. So pretty good numbers of people and animals wearing these GPS devices. And a couple of the animals, a cow, and I think I showed that picture of the dog earlier. Here's our network. This is just showing all kind of animals together, but we actually look at the individual groups of animals in different ways. And here we find that household wealth is a big predictor of time that people are spending in proximity, in overlap with other animals, as well as land size, again, a marker of agricultural wealth. We measure household wealth in a variety of ways. We can just, we do the surveys at people's homes. We can score how the house is built, what it's built of. Is there a concrete slab or is it just on the dirt? Is it wood on the bottom? What kind of roof do they have? And we come up with measures of wealth based on the household. We also have wealth based on goods, and I'll talk a little bit more about that in a few minutes. But anyhow, men have more contact, larger land size, and household wealth predict interactions with other animals based on the GPS data. And so this is only the domesticated animals. The other one included rats and other species. Yes.
The GPS dogs and cats that you have, are those only household animals or there's also cats and dogs out in the community?
These would all be owned animals. They're not feral. Yeah, because we need, we basically find them from the owner. When we survey the owner, we then, find their animals. And if they're willing to, let's sample them. I don't think there are a huge number of feral animals in these, maybe some of the cats, but anyhow, good question. Okay. So, we're finding these are kind of for me, this is the fun thing because we're really learning like what is creating these edges? What are the predictors of those edges? And as you can see, some of these measures of wealth and market integration are predicted. Okay. And people are more likely to interact with dogs, cats, and pigs compared to cattle. If you've met any of these cattle, you might know why. There are not a huge number of cattle, but they are kind of intimidating. What do you think about the cattle, Tom?
I mean, they're not as intimidating as Uganda.
Okay, I'll stay with that. But the other thing that we do not know, the zebu, yeah?
I think they're intimidating. I grew up on a dairy farm, so.
I'm always a little terrified when I'm coming down a trail and one of these animals is coming at me. They also like to interact, they interact more with male animals than female animals. We do not have any idea why that is. Yes. Yes, that's what our statistical models are showing us. But we have not figured that one out yet. So anyhow, those who know Madagascar, please let us know if you have ideas. Okay, so I talked about the ways that we identify infectious disease in these samples. We're actually identifying the organism, but we also have a variety of other approaches that we are using and adopting. This goes a little bit beyond the NIH grant. But it's an approach we're using, which is FIP-seq or phage immunoprecipitation sequencing. VIR-scan, we use this VIR-scan library. It's kind of a high throughput, serology is one way to think about it. And it's looking for, evidence of previous infection with up to 204. I mean, it depends on how you count them, different viruses. So we can get a lot of data, really quickly. Now, the problem is, like a lot of these things, there's a lot of cross-reactivity. You really have to also do confirmatory analyses via serology and other things. And so, we're still trying to figure out exactly how to use this and how to use these data. And we are doing a lot of kind of confirmatory analyses as we go forward with this. We're also doing some other serological tests, including some really interesting results one of my students has on coronaviruses in the region, which I don't want to talk about. I want him to have the pleasure of sharing with groups because I think it's really pretty interesting results. But I'll talk to people later if you want to know about it. But anyhow, really some interesting approaches there. And this is with Lin-Fa Wang at Duke NUS. Duke has a collaboration with the National University of Singapore, Duke NUS, and this is his lab that we've now started to work with a whole bunch of people at Duke NUS, and it's been really productive and satisfying, really great group of people out there. And this has turned up, the VIR-scan has turned up something kind of interesting. It's turned up several interesting things. But the one that we're kind of most actively thinking about is this orth virus. This turned up in a bunch of the samples. It's a zoonotic pox virus, and I'm just going to save you from having to read all this. It's a double-stranded DNA virus. It's typically found in ungulates, especially sheep and goats. It's not known to be transmissible between humans. But this is interesting because we don't have sheep or goats in the system. We're very, very, very few. Certainly we haven't sampled any. And the number of positives is really pretty remarkably high. It's not what you would expect if it's just kind of occasionally spilling over from a cow or a pig or something else. And in fact, it's about 63% of the people for, one of our runs of this are showing really strong evidence for a positive hit for this virus or something like it. And you can't really see this so well because of the lighting, but it's really, according to our colleagues there's very strong evidence that there's something real going on here. Now, as I said, we have to do the kind of confirmatory analyses, but here's where it gets really exciting. I've talked about hookworm and blastocystis. Now we have something new, maybe something that doesn't seem to fit with what, this virus is. And now we have these networks that could help us understand how it might be transmitting. Of course, we have to figure out exactly what it is, but it's, now we have the data to enable us to investigate whether it's environmentally transmitted through close contact, transmitted maybe through, really close contact, deeper kind of social relationships. So it's exciting in a sense, it's never fun to find new viruses in populations, and it is kind of a nasty one, as you can see. It creates these lesions. But we're trying to figure this out and use these network approaches to say something about it. Okay, I wanted to say a few words about a second grant that we're now in year 2 of, again funded through the Ecology and Evolution of Infectious Disease Program, this time through the NSF. This is a multi-agency funding opportunity, as you guys know, and this one's funded through the NSF. And it brings in a few new people. Georgia Titcomb now is a PI, co-PI, Shai Philosoph again, Lin Fa Wang, who I just talked about, Mark Janko, my colleague at Duke Global Health, is doing really amazing work and it's kind of spatial epidemiology and things like that. And it also really brings in the social sciences side a lot more, which I'm excited about, and this idea of market integration. So the first grant really focused on these human-animal contacts, is focusing still, and the human-to-human transmission. And what we're trying to do with this second grant, what we are doing with this second grant, is understanding intercommunity transmission. So this was kind of the logical progression for us, figure out things within communities. Now let's think about between communities, okay? And we have, as I mentioned, these 3 villages. Now, at this point, we have 34 villages, okay? Where we've launched surveys and collected blood spots and collected other samples. And so the 3, highlighted are the ones I was talking about before, but now we really have a good, collection of villages across this region. And it's a great region for doing this because we have Marijeji National Park here. There's like a forest corridor here. And it's really kind of a cul-de-sac. You can only come in, through one road, and there are all these villages off that road. And there's a little bit more of a network of roads down here, but it's really kind of a contained system that's really appealing to us in a lot of ways. Okay, so we ask people in our surveys about their travel history. We asked them to name up to 5 communes or villages, collections of villages within the Sava region that you've traveled to in the past year. And this can go outside of that cul-de-sac I was just talking about, okay? We also asked them to list 5 people who visited them from a different Sava commune. And we collect a blood sample from each subject. And we have, it's like about 3,500 subjects at this point. Actually, this is an earlier slide. It's about 3,550 subjects. And Michelle Pender somehow managed this all from afar. It's really pretty impressive. We also are collecting data on the taxi brew system and how people are moving, and other, just what is their actual mechanism for moving around the region. This is a project that Mark Janko is leading, and, surveying the taxi brew drivers and passengers, figuring out the ways people are moving, how quickly they can move around the region, and also getting biological samples from the animals that get transported, especially chickens, especially poultry. So I'm not going to talk about the taxi brews, but let me talk about the travel survey results, because we have some initial results in now. And again, the hypothesis, that's kind of the overarching hypothesis, is that market integration is influencing the movement of people and infectious diseases around this region. And market integration is this transition from a subsistence-based lifestyle to a market-based lifestyle. And even within a village, you see incredible variation. Some farmers are really, all in on the vanilla and they're buying things, and others are really just, practicing subsistence agriculture, focusing on rice and a few other crops, and often barely getting by. So there's incredible variation within a village, but there's also variation across villages. And I have a slide that'll show that. And so these are our first networks that Tyler's produced from all of this data. This is the whole Sava region. Okay, and again, it's a little hard to see, but you know, a lot of these are the number of participants, fewer to more. We see a lot of major movement out to some of the bigger cities. Okay, the bigger cities in the region, out on the coast or interior in that basin I was talking about. And we see that most of the travel that people are reporting involves for economic reasons, to buy or sell stuff, okay, or to visit family and friends. Up to a lesser extent, it involves healthcare. And again, we can see kind of where those hubs are, and to some extent, school. So we can see right away that economic factors are driving decisions about where people are going and the patterns of movement, as well as, you know, their social connections to family and friends. And this is the slide I thought I had next on market integration measures. So we have the number, here's one measure we use, consumer goods or durable goods. We ask them how many they own. When we ask them a series of items, do you own this? Do you own this? Do you own this? It's like a radio, a TV, a bicycle, you know, things like that that you have to purchase on the market and that are actually kind of expensive, right? And so we get the number of those. You can see there's a lot of variation within villages, but also, you know, some people have zeros on that. A lot of variation within villages and also across villages. Another measure is household construction. Is it a concrete slab? Is it wood on the floor? What are the walls made of? All these kind of things. We have source for more market integration, less market integration, and data on vanilla farming. You know, are you a vanilla farmer? How much do you grow? Percentage of the crops sold, right? Because they're selling them on the market. So that's a measure of market integration. So we have really rich data, you know, from these survey instruments and our own observations of the households. And so the question is, what predicts travel frequency? Now, again, now we have a network that's at the regional level, and we want to understand what predicts those edges on that regional network. And you can see here, it's being male, men travel more than women. Education is predictive. Either secondary or higher versus none is predictive of travel. Having more goods, okay, the household wealth does not predict it. But people have more goods. I guess these are the people who are motivated to travel to go buy those radios and things like that. And also the percentage of crops sold. So it looks like there is this kind of economic motivation to travel, and is predicting, you know, the amount of travel that we see. Now, I don't have infectious disease data yet for you on that, but that's the goal, and that's some of the data that Tyler has on these coronaviruses, and we have all this FIP-seq, the VIR-scan data that's coming in now across these villages, and we basically want to understand, you know, how are these diseases distributed across the region, and can we predict that from the networks that we have, as well as the other social data we have. We published this paper recently, 4 of us on the team, how market integration impacts human disease ecology. So if you're interested in this topic, you know, just to share that, led by my graduate student Lev. And just to kind of come, you know, back full circle here, you know, this is that slide I showed at the beginning. And, you know, I hope I've convinced you. I mean, we certainly don't have all the answers yet, but I hope I've convinced you that this network approach can really help us frame this issue of understanding early stages of pandemics. And we really need to understand not just the local transmission, but the inter-community transmission as well. And just a few take-home messages. We can, you know, I hope I've convinced you that we're generating transmission potential networks that actually predict infection patterns. And we can use those to provide new insights like we did with the hookworm, you know, where it looks like something different is going on depending on the environment in which people are living. And also to test hypotheses for alternative transmission modes. We can include animals in these networks, and that's something we're pretty actively doing, you know, right now, and looking at how that predicts infection. And we can also investigate the factors that influence those social connections. You know, we can say, what are the predictors of these edges, you know, in terms of economic, social factors, demographic factors, right? And then we can scale these methods from within villages to regional transmission, and there also incorporate economic and social factors of this kind of inter-community connectivity. Okay, so I just want to again acknowledge the people who've done the hard work on a lot of these examples I just shared. I want to really recognize our scientific team in Madagascar and of course the people we study. We have a team devoted to the social surveys, to the survey work, to the trapping, and of course the funding that supported this. And thank you guys very much. Appreciate it. Okay, yes. It looks like
I live with like the Tana region, and that, but like what are the sizes of the villages? Like the
Yeah.
northeast area, right? So like what are the sizes of the villages and even like the bigger towns like there in that region?
Yeah, so I don't know so much the bigger cities, and towns. But the villages might range from 300 people, that's about the smallest that we have. And that's adults and children, up to, 5,000,
Okay,
for some of the bigger ones. Most of them are right around 1,500, I'd say is a good kind of median.
and does like the size of the villages like it like impact a lot of that, or is it just like you said, the proximity of the houses rather than the size of the town or village?
Yeah, so the three villages I talked about are fairly similar in size at the beginning. So in terms of the 34 that we now have, we have more variation. And we very carefully chose these villages to capture some that are, small, some that are large, some that are close to the road, because that's going to affect people's travel. Some that are along these taxi Bruce routes and some that aren't. And so there's some of those villages you have to walk for a whole day to get to.
Yeah.
I mentioned that forest corridor and there are 3 of them we've sampled up there and it's pretty far away. And, drive for a whole day and then walk for a whole day. So it's a 2-day journey out to them. So yeah, a lot of variation. And a lot of variation in the market integration measures too.
Yeah.
So it's good. Yes.
So when you choose those villages, so for example, I'm intrigued by the village, case of village.
Yes.
In advance, you choose that village because it looked different from. the
No, we should have been paying more attention.
others. Yes. It looked different already.
Yeah.
So do you think that to conduct a study like that, is it important to take like villages that look really the same, or is it more informative to, actively seeking
Yeah.
for like different patterns?
Yeah, it's a great question. To be honest, the reason why that one's the bias because it was during COVID. So we ended in our first season early and then we couldn't go back to the island. And we kind of did this from afar. And I think if we had gone there in person and seen it, we would not have chosen it because with just 3 replicates, you really don't want one that's different. As it is, 3 is not enough. So we were really looking for 3 similar villages. And it wasn't, it's pretty obvious in hindsight, that it differs. And in fact, as soon as I walked into that village, the first time I was back, I was like, okay, this is a different situation here. So yeah, we were aiming for similarity on those 3 villages. We're aiming for difference on this bigger set of villages. So is that, yeah, okay.
Yes, I know that you mentioned also the distance from the main road.
Yeah.
That's so important.
It is, and we haven't yet looked at that as a variable, but I'm anticipating that it's going to be very predictive. There are some remarkably big villages though that are far from the road. It's kind of incredible to me. Yeah, thank you. Yes. Oh, yes. We have more, we need more, there might be.
So I have 2 questions. One, did you notice any, because you found so many viruses in these people and animals, do you see a pattern when it came to like if one virus disease, or between the worms and the viruses, was there a pattern in terms of like disease occurrence to these individuals? And second, during the time of the study, like what was the environment? Like, was it very humid, rainy? Because that can also impact viral transmission, aerosolization, viral stability, because it can make the material that was more porous.
Yeah.
If trap more water, and that water can help increase biostability. So all these things.
Yeah, so with regard to your first question about co-infection, that's, very much something we're interested in, and we've started to do a little bit of that with the hookworm, the 2 species of hookworm. And there's not really anything super interesting to report there, but yes, we are very much interested in that question and trying to, I'm trying to go at it though with, really clear hypotheses, and like an obvious one might be, the malaria hookworm kind of interaction that people have investigated, but we don't have the malaria data. And so I want to try to go into these with, a priori hypotheses for how kind of interactions might occur. But I don't have anything to report yet, but it's very much of interest to the group. Now, with regard to the other question, we sampled over 3 different seasons, and we're not really seeing a big seasonal effect, for, the organisms that we've looked at in most detail. So there's really not, much to share on that either, but yeah.
Thank you.
Okay, pass on the other hand.
I
Okay.
got a few kind of questions. One thing related to the co-infection question. Stephen Mughal, who was a fellow with the training grant that's associated with this series. He graduated last year, he's an EAS fellow now, but he did some work with us around Ron. So another part of Madagascar, looking at co-infection with malaria and some other parasitic species. So I'm hoping he's gonna be able to come. He couldn't come to your talk, but I'm hoping he'll be able to join us in a half.
Okay, that'd be great. Yeah, I'd like to.
But the question I had immediately was more related to when you were asking people questions about vanilla production. It's a really sensitive topic because of the value and the violence that's been associated with it. So I'm wondering if you ran into challenges with that, with getting reliable answers or different scales of answers relative to other signals of production.
Yeah, that's interesting. We didn't run into that at all.
Okay.
But I should really emphasize, we have a survey team, that are local people.
Yeah.
I mean, they go out there, they live in Indapa, which is in that big basin, but they always work with another person. Not usually from the village itself, and we always interview people, privately.
Yeah.
And so they're not, talking about things in a group. So, but we haven't had any concerns about that.
And then as you've scaled up for the larger scale study that you're doing now, the NSF study, are each of the villages part of distinct Fukutanis, or are there any where there are multiple villages within a given Fukutani?
There could be some that are within the same Fukutani. Yes.
Okay, but then your initial 3 were all from.
Yes.
States. Yeah.
I have to think about that for an about there. I believe so.
Because that.
Now, why do you think that, what is
A lot of times the leadership is at the Fukutani level, so it can influence a number of things, but hopefully we can talk.
Yeah, sorry.
more
Yeah,
about that.
that'd be interesting, yeah. Yeah, I should say, all of this is always done in very close consultation with our survey team.
Yeah.
To really know the region. And with our Malagasy partner, Rural Madagascar and Bahatra Association, who's, an expert on small mammals, but she really helps us figure out where to, it's all done in consultation with them and other local people too. So I look forward to talking more about that. Okay, other questions? Yes.
So, viruses is also very interesting. What can you tell us about why there's such a high seroprevalence for the virus? Where is it coming from? And using VirScan, can you actually distinguish that from seroprevalence and other viruses?
We don't know yet, we don't know where it's coming from. We're really surprised that we get that level of hits. And we're not seeing other viruses that are closely related to it kind of lighting up, on VirScan. Of course, we have to, we're so early in this. This is like just kind of an initial finding. And we're trying to figure out how we can verify this. Because there aren't any we also are finding some other viruses where at least we can get a kit, that could do some testing, to do some confirmatory analyses, but we're having trouble with that with the orth virus. So if anybody studies orth virus or wants to, let us know, because, we're kind of we're hitting some roadblocks. I don't think they're dead ends yet, but
And no other pox viruses,
what's that?
and other pox viruses.
I can't say off the top of my head right now.
But you're not seeing clinical cases, right? You're just,
No.
it's just, you're getting a hit.
That's right. We have not seen any clinical cases. But now we have, because of this result, we're also asking people, in our new surveys, we have questions about this and actually show them pictures of what it would look like on an animal or on people. And so we're going to be able to, and, we obviously didn't know we were going to find it, or we would have asked before, but that'll also help us to, see if we're seeing positives in people who've reported it on themselves or things like that. It's just another, it's still not going to confirm what it is, but it's going to confirm that there's some virus that's like this. I mean, it could be something totally unknown. It could be some, kind of cross-reactivity, but we've literally just got these results in December. We're trying to figure out what to do. So seriously, if somebody is interested in this virus, let us know. Yes.
I actually have questions related to the orth virus. So in terms of the communities, is it typical that someone that is born in a village stays in that village, or do they have a tendency to move between villages? Because it may be a historical expansion where it's not in the current village.
Right.
Experience for everyone.
Yeah. Well, there is a lot of movement,
Okay.
and I mean, yes, I think, on average, people are staying, but they do move around the region some. And of course, there have been migrants into the region. And we don't necessarily know from our surveys whether that's, the case for particular people. But, I mean, I'd say on average, they're pretty stable communities. I mean, we go back year after year and certainly see the same people, but we know from talking to people that they describe, oh, I grew up in that village. There's a very common, kind of discussion point. And so there is at least some local movement of a lot of these things. But I can't give you a number.
No, that's fine. And then follow up, since you have serum sample, you can actually look at the isotype of the antibody. So it's an IgA antibody, is probably a relatively recent exposure, which is IgA. So since you have those serum samples, you can look at the isotype for the orth virus to determine if it's recent or historical.
Yeah, that's interesting. We have dried blood spots.
Oh.
Filter on 903 cards, which is, for stabilizes proteins. So I don't know, and we don't have a lot of blood either. So that's been a limitation as well. But yeah, do you think you could do that off of 903 cards?
Depending on how old samples are.
Yeah. So we just don't have the refrigeration or, those kind of things we do get some serum, but it's very limited. And we have to be very strategic about it to get it in the freezers. Okay, yes.
It's while we're talking about pox viruses.
Okay.
Have you been looking at the distribution of the seroprevalence for this orth virus with age? Do you see when it starts popping up? And I know you've only looked at adults, but do you see any sort of like?
I'm trying to think back to the very initial analysis one of the graduate students on the team did, and she was not finding an age effect as far as I remember. But I'd have to refresh my memory to be 100% sure. So we have not done any of the network analyses, to see if there's any kind of connectivity amongst the cases, for environmental overlap, household overlaps, those kind of things. So.
I know there's a lot more questions, but I want to give people an opportunity to get to their other things, but you're gonna be here for a while. I know there's gonna be more meetings and dinner and stuff. Yeah.
Yeah, I'm happy to talk to people for a while afterwards.
Thank you very much.